Anur Naja. I'm from Bachelor in Nutrition. So I'm gonna talk about the introductory of obesity. What is the definition of obesity? So obesity is actually, I think everyone has an overview about what obesity is, but the real definition to it is actually obesity is a medical condition in which excess body fat has accumulated to an extent that it may have a negative effect on health. I think most of us are familiar when it comes to how obesity or the excessive fats in us that can cause diseases. Obesity is a serious disease up to a point where it could be considered as a major public health problem. Why do I say this? Is because the percentage of obesity around the world increases year by year, and it is not only because of the excessive fats that is dangerous for the humans itself, but obesity increases morbidity risk for heart diseases, type 2 diabetes, and some type of cancer as well. Metabolic changes that could lead to these diseases in the body should be warned about, such as abdominal obesity, elevated triglycerides, reduced high-density lipoprotein, cholesterol, high blood pressure, and elevated fasting blood glucose. If any of us have these three conditions, out of all this, we are considered to have obesity, and it's really dangerous because it could cause you these harmful diseases. Lipids are key macronutrients in the human diet. Lipids are important for our life, but the type and proportion of dietary fatty acids consumed impact health and whole body physiology. So everything that we take, it has to be taken in a small amount and an appropriate amount. So, research has shown that saturated fatty acids are detrimental to health. What I can say is, such as fast food. Fast food contain a lot of fats, and they are not good for our health because it's a processed food. While monounsaturated and polyunsaturated fatty acids offer health benefits. So in this case study, in this presentation, that we're going to talk about how these monounsaturated and polyunsaturated fatty acids, like omega-3 fatty acids, as well as fish oil or fatty fish in omega-3, such as eicosapentaenoic acid or docosahexaenoic acid, have demonstrated cardioprotective, anti-inflammatory, and hypotriglyceridemic properties. Hence, these fatty acids assist in the treatment and prevention of obesity comorbidities. In this review, we are going to talk on how the effects on omega-3 profiles on obesity, as well as metabolic changes in both animal and human studies. It is also highlighting the potential mechanisms for omega-3 in reducing body weight, improving body composition, and counteracting the adverse metabolic consequences of obesity.
Hi, my name is Darshini and I'm studying for Bachelor in Nutrition, and today I'm going to present about adipose tissue and obesity. So first of all, we need to know what is adipose tissue. Okay, actually the adipose tissue is acts like an insulating layer that helps to reduce heat loss through our skin, and also a protective function that providing a mechanical protection like padding to support around some major organs like kidney and etc. So the body fat is primarily stored at adipose tissue, and is also known as a energy storage, and this connective tissue is composed in five actually. So first is adipocytes, second is pre-adipocyte, third is vascular endothelial cells, fourth is fibroblasts, and fifth is various type of immune cells. So first we go through with these cells. So what is adipocyte? So adipocyte is a cell that specialized for storage actually, and fat that can be found in the connective tissues actually, and important of body in maintaining the proper energy balance, and then storing like energy source in response of hormonal stimulation, and then second is the pre-adipocytes. So pre-adipocytes is like undifferentiated fibroblasts that can be stimulated to form an adipocytes actually. And then number three is vascular endothelial cells. It's also known as a thin membrane actually that lies with inside the heart and the blood vessels. And then number four is fibroblast. So fibroblast is like a connective tissue which produces the collagen and other fibers. And then number five is like a various type of immune cells, which is we known as lymphocytes, which is T cells, B cells, and NK cells. And this adipose tissue, okay, actually is an active endocrine organ, and that secretes numerous hormone, like including leptin and adipotin. So what is leptin and adipotin? So leptin is a hormone predominantly like made by adipose cells that helps to regulate energy balance by inhibiting hunger. And then second is the adipotin. So adipotin is a protein hormone, okay, that includes or involve, okay, and regulating a glucose level as well as fatty acids breakdown. Okay, and then we go for the next slide. So the next slide is we go for 2.1, which is three major types of tissues. So what are these three major types of tissue? Is like white adipose tissue, brown adipose tissue, and beige adipose tissue. So this white adipose tissue is also known as white fat, and for non-weight man, white adipose tissues like it's like composes 20% of body weight in man and 25% of body weight in woman. So this like resulting the storing excess of calories. So when we consume many calories in the body, so the body converts them, which is the calories, okay, the calories, and convert them into the energy in the form of white fat actually. And then we go for the brown adipose tissue. So brown adipose tissue is also known as a brown fat actually, and the main function is to turn food into the body heat, like a thermogenesis. And when we compare this white adipose tissue and brown adipose tissue, white adipocytes are like have single lipid droplet actually, but brown adipocytes contain many small lipid droplet as a high iron containing mitochondria, and it is a high iron content that gives a brown fat its dark red to the tan colors. Tan means not number, is a suntan, is a type of colors like tan colors. And then we go for number 3, which is beige adipose tissue. So beige adipose tissues like beige like cells that develop white fat and then a response of the various activators and reduce the metabolic disease, including the obesity actually. And then we go for 2.2 obesity. So 2.2 obesity, we also it's known as the obesity that leads the adipose tissue it's like a dysfunction actually, the weight loss like via the lifestyle modification is the primary treatment in the management of the obesity. So the last part is a treatment. So the treatment we have divided into 3. So the obesity can be treated, there's no worries. So how they treated by 3 types, which is like pharmaceutical, surgery, and dietary supplements. So pharmaceuticals, they can take type of medications actually, got a lot of medication in pharmaceuticals. So for examples like lorcaserin, Orlistat, rimonabant, metformin, and extra. And then for surgery, most of the weight loss surgery that doctors do is a laparoscopic surgery. It's the very famous, and they usually do that surgery for the obesity patients actually. And then number 3 is a dietary supplement, which is the last part, is a dietary supplement. So dietary supplements, they can take as caffeine, and then hydrosiod, raspberry ketones, and then a green coffee bean extracts, and etc. So obesity can be treated, but not only we can take this type of pharmaceutical surgery or dietary supplements, we also can maintain our diet, and we can do our regular exercise with a doctor consultation, and we can cure this obesity disease as well, as we know, and we have to control our food. That's it. So these are my presentation, and thank you for listening. Thank you.
Okay, now I have responsibility to present about omega-3. Okay, omega-3 consists of fatty acid. Fatty acid is a carboxylic acid consisting of a hydrocarbon chain and a terminal carboxyl group. Omega-3 is good for our health. 4.3.1 suggestions about synthesis and metabolism. Omega-3 also provide form from sunflower oil, then corn, vanilla, and soybean. Nowadays, we can form omega-3 on capsule that sell on pharmacy. The easier to get that. 4.3.3 about dietary omega, obesity and metabolic disorder. Omega-3 also increase fat in the body that is particularly suitable for people in the arctic region. For example, Japan and Eskimo businessmen need to get a lot of omega-3 supplies because they are stay at a low temperature places. With that, they have attribute to get more fatty fish intake. Then they contribute to get more fatty fish intake because they place the temperature and have more build body fat. Because omega-3 is a good body fat for us. Furthermore, taking omega-3 can increase the good fats in our body and get energy surplus and strengthen our heart. What can be related with omega-3 and food technology is can research about energy, how to make omega-3 become better and better nowadays, and have a build capsule and can analyze the required atom and raw metal to study with omega-3. And that's all from me.
Assalamualaikum to Dr. Zak. My name is Nur Amadini binti Azizan and I'm a BFSC student. Today I'll be presenting about the foods that are fortified with omega-3 fatty acid. There are various of food that are being fortified with omega-3 fatty acid, but the one that I'm going to be explained is about eggs and also instant food. So in process of making the omega-3 eggs, it started with the diet. The hand will be fed with a food that is rich in linseed, flaxseed, or any corresponding oil, and therefore it causes the enrichment of the yolk with alpha linolenic acid, or known as ALA, and also the enrichment of docosahexaenoic acid, known as DHA. So the eggs that are derived from the ALA fat have a higher in content, while the one that is derived from the hand that being fed with the DHA will have a higher DHA level. However, in some cases, the arachidonic acid levels are being reduced, and therefore by modifying it is subsequent metabolism into eicosanoids believed to be targeted effects underlying some of the benefits of the N3-PFA. The benefits of eating omega-3 eggs will help in increase in systolic and diastolic blood pressure, and also it will help in decreasing the plasma triglyceride concentration. So that's basically about the eggs. Now I'm going to explain about the other example, which is instant food. During instant food, the micro encapsulation will help to stabilize the fish oil against the oxidation, and will make it possible to transform the oil into a powder form. It occurs when at least droplets of oil surrounded by a dry matter of proteins and or carbohydrates coating materials, and then the micro encapsulated fish oil powder enables the fortification of the instant food due to the pressure of the oxygen. The presence of the oxygen will strongly reduce the sensory quality of the fish oil. And as for the flavor, the presence of the flavor allows the higher level of fish oil addition to instant food to conceal the undesirable flavor. So that's all from me. Thank you.
Hi, my name is Christina Osman. I'm from the NT course. Today, my topic, I will be talking about the relation of omega-3 PUFA, which is the polyunsaturated fatty acid, and weight loss in humans. So the investigation on this topic was done to see the effect of omega-3 PUFA on the body weight, body composition, and the energy intake in human body, with also different types of fish and fish oil content, particularly EPA and DHA. It can also be It can also be observed from table 1.0 that the condition of participant can be ranged from healthy to obese. Also, some of the participant has a health condition such as type 2 diabetes mellitus and postmenopausal. The control experiment was done so it can be compared by the other studies. It contained assorted oils containing omega-6 PUFA. One study in health adults consists of fish oil diets and dietary intake controlled, shows results in decreasing body fat mass, basal respiratory quotient, and increasing basal lipid oxidation. Another study shows result in decreasing in fat mass, increased significantly lean mass, despite no alteration to total body mass, RMR, resting metabolic rate, and respiratory exchange ratio when compared to the placebo supplementation, which is the control experiment. Participants also reported diet diaries that show significantly decreasing in carbohydrates, fats, and total calorie intakes, with no change in energy intake and body weight, with only omega-3 PUFA supplement alone, as it can be observed in table 1.0. Next topic is about the omega-3 PUFA in combination with dietary intervention. Results appear more promisingly in weight loss with the combination of omega-3 PUFA supplementation with calorie restriction, as it can be seen in table 2.0. The combination of the two gives greater improvement in insulin resistance and decreasing the triglycerides, rather than taking the calorie restriction alone or replacement of SFA. By the observation from table 2.0, it can be concluded that weight loss benefits may not depend solely on variations in omega-3 PUFA dosages. With low-calorie diet, it gives rapid weight loss result. In table for topic 5.2, you can see the table 3. You can observe from the table 3.0 the influence of omega-3 PUFA plus with exercise and with or without dietary intervention, which consists of the nutritional counseling rather than prescribed diet. The result, as can be seen in table 3.0, gives greater weight loss. Also, the influence of omega-3 PUFA with exercises and with or without dietary intervention prevent valid comparisons across studies because of the difference in intensity and forms of exercise, like aerobic exercise with addition of omega-3 PUFA without dietary intervention gives result in decreasing fat mass. Also, like resistance, other example like resistance training, also with addition omega-3 PUFA and without dietary intervention gives result in increasing lean mass plus improved muscle quality. Lastly, the 5.3 topic, which is limitations in human studies. The findings on the effects of omega-3 PUFA in humans are inconclusive. Improvement in study design and analysis could help resolve apparent inconsistencies in the effect of omega-3 to weight and body composition, and also gender, metabolic phenotype, geographic location, and the type of omega-3 supplementation. It can be concluded that omega-3 PUFA consumption is parallel to omega-3 PUFA concentration in the body. And that's all from me. Thank you.
Hello, my name is Bushra. I'm going to explain two mechanisms by which omega-3 polyunsaturated fatty acid improve adiposity, and metabolic disorder. So adipose tissue is composed of specialized cells called adipocyte. In obesity case, adipose tissue expands through enlargement and increase in number of adipocytes due to a process called adipogenesis. Adipogenesis is a process at which pre-adipocytes become mature adipocytes. Polyunsaturated fatty acid, particularly arachidonic acid, and its metabolites bind to regulate peroxisome proliferator activated receptor gamma and delta in order to increase in expression of adipocyte gene, including lipoprotein lipase. Increased concentrations of omega-3 and omega-6 polyunsaturated fatty acid in human subcutaneous tissue is associated with reduced adipocyte size, whereby increased saturated fatty acid concentration lead to increased fat cell size. Differences in fatty acid concentration are associated more strongly with abdominal subcutaneous than visceral adipose tissue. So studies performed in 3T3L1 cells, which are clonal adipocytes, also shows upregulation in PPAR gamma expression, adipogenesis, and lipid droplet formation after omega-3 polyunsaturated fatty acid has been added. In conclusion, omega-3 fatty acid promotes adipogenesis and healthy expansion of adipose tissue during positive energy balance and promoting a metabolically healthy phenotype. So the second mechanism is adipose tissue inflammation. Obesity causes low-grade inflammation, which contributes to systemic metabolic abnormality that is associated with disorders related to obesity. Adipokines have pro-inflammatory and anti-inflammatory activities, and uncontrolled production or secretion of adipokines due to adipose tissue abnormality can contribute to development of obesity-linked complications. So omega-3 fatty acid blocks the function of nuclear factor kappa B, which is an essential transcription factor for a maximal expression of many cytosins that are involved in causing inflammatory disease. Cytokines like IL-1, IL-6, and TNF-alpha are all elevated in obesity. So omega-3 polyunsaturated fatty acids act as an agonist for different members of free fatty acid receptor family located in a variety of cell types involved in energy homeostasis and inflammatory response. Agonists in stimulation that blocks the inflammatory responses occur through activation of G protein independent signaling pathway through interaction with beta-arrestin proteins and further interacts with transforming growth factor kinase protein, which is TAK1, and binding protein TAB1. Stimulation of free fatty acid receptor 4 or B-arrestin inhibits polysaccharide as it causes the release of inflammatory cytosine like TNF-alpha and IL-6 in a macrophage-like cell line called RAW2647. This is an Abelson leukemia virus transformed cell line derived from BAB mice. Also, omega-3 polyunsaturated fatty acid anti-inflammatory effects are generated through free fatty acid receptor 4 activation, resulting in decrease of macrophage infiltration. Omega-3 polyunsaturated fatty acid supplementation causes M1 macrophage present in adipose tissue to be reduced and therefore decrease pro-inflammatory markers like IL-8. Omega-3 stops inflammatory processes by inhibiting activation of NLRP3 inflammasome by arrestin free fatty acid receptor 4 dependent pathway, which excites a caspase dependent cascade, resulting in release of pro-inflammatory cytosine. The omega-3 polyunsaturated fatty acid DHA acts through free fatty acid 1 and free fatty acid receptor 4 to inhibit caspase 1 activity via formation of beta-arrestin 2 NLRP3 or NLRP1B complex, and thereby decrease pro-inflammatory cytosine. Thank you.
So I would like to explain to you about the function of the N3 polyunsaturated fatty acid or the omega-3 fatty acid in the function of appetite suppression. The N3 polyunsaturated fatty acid can also act as endocannabinoids that will target the hormone that are involved in food intake and the energy signaling pathway. The endocannabinoids can act as a modulatory system in the function of the brain and the endocrine system, which will influence our hormone production. And the modulator function is, for example, it acts like an antenna or a signal transmission, or in this case, a substance that which will indirectly influence the effect of the primary glands that directly activate or deactivate the function of a targeted protein. An experiment was conducted, and it is shown that people that are consume the higher amount of the omega-3 fatty acid content meal, for example, like fatty fish and also fish oil, for example, like tuna or salmon, which has a very high fat content, it is shown that they have the increase in sensation of fullness than those that are consumed lower omega-3 fatty acid contain meals. For example, like in lean fish that has very low fat and very high muscle content. And the realized feeling of hunger and the increased feeling of satiety could aid in the weight loss. This is because due to the number of food intakes which has been reduced. And the polyunsaturated fatty acid, the N3 polyunsaturated fatty acid, can act as an agonist, which means it will help with the secretion of cholecystokinin, which is a peptide hormone that will synthesize in the liver and will be released in the small intestine and has a role in hunger suppression. So with the increased amount of cholecystokinin, it will also reduce the feeling of hunger. Okay, so we explain about the lipid metabolism for the polyunsaturated fatty acid supplementation. Okay, during the omega-3 polyunsaturated fatty acid supplementation, the reduction of weight or fat mass were not accompanied by the change in the energy intake. The omega-3 polyunsaturated fatty acid can split up the energy in the dietary energy, which is the energy that we gain from eating, away from the storage, and it will be used for oxidation by suppressing the lipogenic genes and will activate the genes that encode for mitochondrial and fatty acid oxidation in both the liver and the muscle. This is because the omega-3 polyunsaturated fatty acid has a cardioprotective properties, which means it will protect everything, it will protect the heart from any illness or dysfunction. And it can also affect the endothelial function in the patients. And the endothelial function is referring to the endothelium, which is the cell in the innermost layer of our blood vessel. And during exercise, the consumption of fish oil has been shown to increase the artery dilation and the blood flow to the skeletal muscle. Hence, it will improve the blood flow and will also increase the delivery of fat to be utilized as energy in the skeletal muscle. This is especially during exercise. And the polyunsaturated fatty acid can also limit the amount of fatty acid delivers to the muscle and the liver. And the omega-3 fatty acid has also shown to increase the mitochondrial biogenesis, which is the processes for the mitochondria to enlarge in size, and the fatty acid oxidation in the liver, adipose tissue, and the small intestine. So I will explain next about the function of the omega-3 fatty acid in its role to reduce the insulin resistance. The adipose tissue inflammation is partly responsible for obesity-associated insulin resistance. And since previous research showed that the polyunsaturated fatty acid, the omega-3 fatty acid, are also capable of reducing the adipose tissue inflammation, thus it will help to reduce the insulin resistance within the body. And the hepatic insulin resistance is a characteristic of, is an example of a metabolism dysregulation which is commonly seen in patients that has obesity and patients that have a type 2 diabetes mellitus. And the omega-3 fatty acid supplementation can prevent the insulin resistance in the muscle, which is partly by improving the glycogen synthesis. And the omega-3 fatty acid can also help to decrease the fat content in the muscle and maintaining the transcription of the glucose transporter type 4, which will help improve the myotubular glucose intake and reducing the muscular insulin resistance. Okay, so the myotubular glucose intake I will explain the myotubule. Myotubule is a developing skeletal muscle fiber which has a centrally located nucleus and one of the three major muscle in the human body. And with the increased intake of the glucose for the myotubule, it will have enough energy to develop into a muscle, and this can also prevent the muscular and the hepatic insulin resistance, which is associated with obesity. However, the polyunsaturated fatty acid, the omega-3 polyunsaturated fatty acid, is ineffective when the patient is confirmed with the type 2 diabetes mellitus, but will be effective for patients that only have the obesity.
Hi, my name is Moira Shikin, and I'm currently taking BFST. And first of all, I'll be talking about liver X receptor in lipid metabolism. Okay, in liver X receptor, nuclear receptors that function as liver X receptor are nuclear receptors that function as cholesterol sensors and regulate cholesterol homeostasis and indicate as role in inhibition of liver X receptor alpha in reduced sterol regulatory element binding, expression with N3 PUFA, which is polyunsaturated fatty acid, but it depends on the cell types. So sterol regulatory element binding, 1C, synthesized, transport, and maturation are increased with insulin. As for polyunsaturated fatty acid, prostaglandins and leucorrhias can all act as ligands for peroxisome proliferator activated receptors. Okay, peroxisome proliferator activated receptors transcription factor that form heterodimers with retinoid and receptors in the promoter regions of several genes involved in lipids and glucose metabolism. For example, entry PUFA activation of peroxisome proliferator activated receptors alpha decreases lipogenesis by suppressing FAS activity. As for omega-3 PUFA or polyunsaturated fatty acid, increases mitochondrial biogenesis and fatty acid oxidation in the liver, adipose tissue and small intestine of rodents, possibly through PPAR alpha or peroxisome proliferator activated receptors alpha and COX-3.
Hi, my name is Noor Aida Shakira. I'll be talking about thermogenesis. Thermogenesis is a process where heat is produced. This is examined by cold and diet-induced thermogenesis, which was mediated by mitochondrial uncoupling proteins, UCPs, in the presence of N3 PUFA, polyunsaturated fatty acids. UCPs are inner mitochondrial proteins that transport hydrogen ions across the mitochondrial inner membrane. In the journal that I read, they have shown that brown adipose tissues, BAT, from EPA supplemented mice shows higher levels of thermogenic genes, such as PRDM16, which acts as a transcription co-regulator that controls the development of brown adipocytes. Omega-3 PUFAs increased mitochondrial oxidative capacity in white adipose tissue, WAT, and skeletal muscles. Most studies were carried out at 20 degrees Celsius, which caused the results to be unclear whether the increase in mitochondrial oxidative capacity were caused by the N3 PUFA induced or the cold induced. And there was no big difference in body weight, but there was a decrease in epididymal fat mass. This is with high fat diet supplemented with N3 PUFA mice at 30 degrees Celsius. This also indicates that N3 PUFAs could reduce body fat accumulation even at thermoneutrality and independent of cold-induced thermogenesis. There should be more research done for the mechanism of the role of N3 PUFA. Next in the article that I read was about lean mass. N3 PUFA has the potential to increase lean mass. It is not yet fully understood, but there is a possibility that it includes both catabolic and anabolic pathways. The increased lean mass results in improved body composition and possible improved metabolism, even though there is an increase in resting metabolic rates has been demonstrated with the increase of lean mass, but post N3 PUFA are not always accompanied by the increase in RMR. Omega-3 PUFAs have shown to lead to muscle protein breakdown in isolated muscles of the mice. Increases in protein synthesis may cause dispute between N3 PUFA's activation of the protein synthesis signaling pathways. This is a key pathway in muscle growth. The increase of the whole body protein turnover under insulin stimulation, but no significant increase in lean mass. Shown in the following 9 months of the N3 PUFA supplementation, changes in the protein dynamics may not translate to increase in protein mass. One possible mechanism of N3 PUFA is the increase in lean mass in the alteration of protein dynamics, which is related to N3 PUFA anti-inflammatory properties. There is also another possible mechanism, which is the ability to lower cortisol levels. Epigenetics is an important contributor to many chronic diseases, including obesity. Limited studies have examined N3 PUFA and epigenetics modification, even though the expression of several genes involved in metabolic homeostasis is regulated by DNA methylation. In a population-based study in Alaska, A few studies reported that fish oil supplementation did not alter the methylation pattern of genes, including leptin and the leptin receptors in mouse epididymal flaps. This may be the N3 PUFAS works through epigenetic mechanisms other than methylation. MicroRNAs, short non-coding RNAs, which act as post-transcriptional regulators of genes, which act as sequence-specific inhibitors of mRNA. It has been documented that mRNA act as key regulators in the pathogenesis of metabolic disease by affecting inflammation and lipid metabolism.
As for future perspectives, both animal and human studies have examined the beneficial effects of combining N3 polyunsaturated fatty acids with other dietary supplements and pharmaceuticals, including anti-diabetic drugs, L-alanine L-glutamine, as well as alpha-lipoic acid and krill oil. Animal studies using the combination of N3 polyunsaturated fatty acid and rosiglitazone have reported a significantly greater reductions in body weight, enhanced oxidation of fatty acids, and counteraction of lipogenesis than with rosiglitazone therapy alone. Lastly, omega-3 polyunsaturated fatty acid supplementation in addition to anti-diabetic pharmaceuticals could attenuate body weight gain caused by pharmaceuticals and should be further investigated. Thank you, that's all.
Hello, I'm Sarah. I'm taking Bachelor in Nutrition and I'm going to talk about PUFAs modulate adipokine secretion. Obese individuals have high plasma leptin levels suggestive of leptin resistance. Conversely, weight loss leads to parallel decreases in plasma leptin levels, and this weight loss associated decrease in leptin could contribute to hunger and lower metabolic rate and ultimately lead to weight regain. So EPA supplementation attenuates the decrease in blood leptin levels that occurs during weight loss in obese women, suggesting a potentially significant rate of EPA and weight loss maintenance. Indeed, EPA increases the production of leptin in rodents and cultured adipocytes, suggesting a direct effect of N3-PUFA on leptin production. However, the few studies that have assessed the role of N3-PUFA in weight maintenance have found no significant effect on weight or blood leptin concentrations between N3-PUFA supplemented subjects compared to other weight loss groups. So omega-3 PUFA mediated effects on leptin are dependent on a number of factors, such as diet, composition, and energy balance, which could cause conflicting results. Both animal and human studies have found significant increases in blood levels of the insulin synthesizing adipokine, adiponectin, following N3-PUFA concentration. EPA appears to regulate adiponectin levels at the translational or post-translational level rather than at the transcriptional level. It has been proposed that the anti-inflammatory properties N3-PUFA supplementation induce an increase in adipocyte adiponectin production and improve leptin sensitivity. And this type of interplay could have a significant influence on body weight regulation. An inverse relationship between serum adiponectin concentrations and TNF-alpha has also been demonstrated in obese and in overweight and insulin-resistant children following N3pUFA supplementation. Fatty acid-binding proteins are cytosolic proteins that bind long-chain fatty acids and promote transport to several organelles. Fatty acid-binding protein 4, FABP4, adipocyte FABP, A, FABP, or AP2 is secreted from both macrophage and adipocytes and functions as an adipokine. An elevated FABP4 serum concentration is associated with obesity, insulin resistance, and hypertension. Adipocytes are the predominant contributors of circulating FABP4, and during lipolysis, FABP4 functions in a non-classical secretion pathway. However, omega-3 PUFA does dependently reduce FABP4 secretion in 3T3L1 adipocytes and reduced serum FABP4 concentrations in human. Omega-3 PUFA mediated reductions in FABP4 could also be due in part to reduced expression of transcription factors involved in adipocyte differentiation, including PPARY2 and CEP. Another possible mechanism by which FABP4 levels are lowered by N3pUFA is through the beta adrenergic receptor. Since N3pUFAs reduce sympathetic nerve activity and thus may lower FABP4 serum level. Taken together, N3pUFAs modulate adipokine secretion by exerting anti-inflammatory effects and promoting a metabolically healthy phenotype. That's all from me. Thank you.